Professor Shinya Yamanaka, it is a great pleasure to be able to welcome you to Uppsala University, and we are truly honored that you take the time to come here and give a lecture. Professor Yamanaka got his MD at Kobe University in 1987 and PhD from Osaka University in 1993. He then went for a postdoc to the Gladstone Institutes in San Francisco, and after returning to Japan, he took up a position at Osaka City University, and in 1999 was appointed associate professor at Nara Institute of Science and Technology and a full professor in 2003. He is now a professor at Kyoto University and since 2008 the director of the Center for iPS Cell Research and Application. He maintains a position as senior investigator at the Gladstone Institutes in San Francisco. He's been given numerous awards, including the Lasker Award, and his research has been focused on how is pluripotency of stem cells retained, and he's moved over the last number of years to explore even more far-reaching ideas, such as can pluripotency be reinstalled? Can a mature cell, by forced expression of a certain genes, be forced to go back in time to an embryonic stem cell state? And we know now that the answer is yes. And I think when we read the first iPS paper in Cell in 2006, it was the kind of paper that made your thoughts shifts from, wow, this is not possible, to, wow, this will have a tremendous impact on biology and medicine. And I'd like to quote from the final statement of the Cell paper discussion, which says, "The finding is an important step in controlling pluripotency, which may eventually allow the creation of pluripotent cells directly from somatic cells of patients." End of quote. And as we know, this followed shortly, and we will hear from your lecture about this. And across the world, iPS research took off, and we have already seen its impact on science, in particular in disease modeling. But we're still in the dawn of induced pluripotent stem cell possibilities for medicine. There is so much more to learn, and among our responsibilities as scientists is the task to balance expectations while keeping enthusiastic about future therapies. So please join me in a warm welcome for Professor Shinya Yamanaka.
So thank you very much, Professor Forsberg Nilsson, for your very kind introduction. And thank you very much for inviting me to this beautiful campus. Our emperor came to this university to receive an honorable degree a few years ago. So it is a great, great honor for me to be here. In my talk today, I would like to discuss two issues. The first one is researches of myself and by others that led to the generation of iPS cells, induced pluripotent stem cells. Then I would like to discuss the potential of iPS cells. So let me start with my earlier days. I started my career as an orthopedic surgeon 25 years ago. But it didn't take long for me to realize that I was not talented at all in surgery. So I gave up and I decided to change my career to become a scientist. I got my PhD in Osaka, Japan, and then I did my postdoctoral training in San Francisco at Gladstone Institute of Cardiovascular Diseases. This is my then mentor, Dr. Thomas Inerarity, and myself. I believe this picture was taken in 1991. The purpose of this slide is to show you that I used to have more hairs. So this is what I worked during my postdoc training. At that time, Dr. Inerarity was interested in one gene, one protein, ApoBec1, which plays an important role in the metabolism of cholesterol, plasma cholesterol. So this is his hypothesis. He thought by overexpressing this gene, ApoBec1, in liver, that would lower plasma cholesterol levels, and that would prevent atherosclerosis. Well, you know, I was in Gladstone Institute of Cardiovascular Diseases. To test his hypothesis, this is what Dr. Inerarity, asked me: to make transgenic mice expressing ApoBec1 in a liver-specific manner. So I worked very hard, day and night, and I was able to establish mice, liver-specific ApoBec1 transgenic mice very quickly. One day in the morning, a technician working with me, taking care of my mice, rushed to me and said something very strange. She said, Shinya, many of your mice are pregnant, but they are male. So I got confused. I went to the animal facility to see what is going on, and this is what I found. So instead of many babies, I found this cancer in liver. So it turned out that this gene, ApoBec1, is a potent oncogene. We can never use this gene to prevent atherosclerosis. It's kind of a crime. So this is a very unexpected result. Very naturally, Dr. Inerarity got disappointed. But I got very, very excited by this unexpected result. I was very interested in why in the world we got this cancer by this ApoBec1 gene. And also Tom, as a scientist, Dr. Inerarity got excited too. So he allowed me to work on this phenotype. So I became the only one researcher who worked on liver cancer in Gladstone Institute of Cardiovascular Diseases. In the following few years, I did my best to understand what's going on in these livers. And I was able to identify one new gene, which I designated NAT1, novel ApoBec1 target number 1, very simple name. So I identified this gene as a candidate responsible for ApoBec1 mediated cancer. From its protein sequence, I thought NAT1 is a function as a tumor suppressor gene. And by overexpressed ApoBec1, NAT1 function is kind of destroyed. So I thought, well, oh, this must be the mechanism. In order to prove my own hypothesis, I generated knockout mice of NAT1 and also knockout ES cells, embryonic stem cells, deficient in NAT1. And again, I observed totally unexpected result. It turned out that NAT1 is essential for pluripotency of mouse embryonic stem cells, ES cells. So as you all know, ES cells were derived from mouse embryos, inner cell mass of blastocysts in 1981 by Dr. Sir Martin Evans and also by Dr. Gail Martin. ES cells have two important properties. The first one is their immortality. We can expand the number of ES cells as much as we want. They can proliferate almost indefinitely. The other important property is pluripotency, the ability to differentiate into virtually all types of somatic and germ cells that exist in mouse body, including like neurons, muscles, and so on. And it turned out from my research that NAT1 is indispensable in pluripotency. You know, I generated NAT1 now, ES cells, they can proliferate normally. However, when I try to induce differentiation of NAT1 now ES cells, they cannot. So from this unexpected result, ES cells became my research target. Before this result, ES cells was just a tool to make knockout mice. It was just a tool. But because of this, NAT1 became, I'm sorry, ES cells became my research target. So when I was working on NAT1, we went back to Japan. You know, I wanted to stay in United States as long as possible, but because of many reasons, like family reasons, I decided to go back. And I became sick. I suffered from PAD. So there should be many real doctors in this room, but you may wonder what PAD is. Well, actually, I coined the word. PAD stands for post-America depression. So, you know, I had a wonderful time in San Francisco. You know, I was able to concentrate on my science, and you know, there were many talented, many good researchers around me, including several Nobel laureates. And they often, you know, told me, well, Shinya, your result is very interesting. Your science is very interesting. Keep going on, something like that. But as soon as I went back to Japan, I lost that kind of environment. Nobody was able to understand what I was working on. I was often told by my colleagues, Shinya, you know, those cells, mouse cells, may be interesting, but I really think you should do something more related to human patients. So it was, and also I had many mice. And I had to take care all of them all by myself, twice a week. So because of those reasons, I got depressed. And so badly that I was about to quit from science, escape from science. But very luckily, two events happened in my life which rescued me from PAD. The first event was the generation of human ear cells in 1998 by Dr. James Thomson. He published a paper in Science. I got very excited because human ear cells have the two important properties as mouse ear cells. We can expand human ear cells as much as we want. Then we can make many types of human cells in a large quantity, including dopaminergic neuron, neural stem cells, cardiac cells, pancreatic cells, hepatic cells, and so on. Then, by transplanting these human cells, we should be able to treat patients suffering from various diseases and injuries, including Parkinson's disease, spinal cord injury, cardiac failure, diabetics, and so on. So it turned out that ES cells, the cell I was working on is very, very related to human medicine. I may be able to contribute to human patients by working on ES cells. So this is one reason why I was able to recover my PhD. The other event is my promotion. In 1999, I became a principal investigator in Nara Institute of Science and Technology. As you can see, this institute has a beautiful campus. Not as beautiful as this campus, but it's good enough for me. And they have many, many young faculty members. They have good equipment, and most importantly, they have many young, talented, hardworking PhD students. So because of these two reasons, I was able to overcome my PhD, and I was able to continue my research. Because I got my own laboratory for the first time, I thought I should have some clear vision or clear goal of my lab to attract students. So I made this as a long-term goal or vision of my own lab. So I said to my the first year students, this is my goal. I want to make ES cells, not from human embryos, but from somatic cells like skin cells. I thought this goal was very important because human ES cells has a very high moral or ethical hurdle. Many people are still against the usage of human ES cells because we have to use human embryos to generate human ES cells. But if we can make ES-like cells, not from embryos, but from patients' own cells, we can overcome this hurdle. So I thought we can achieve this goal by identifying reprogramming factors that can induce pluripotency in somatic cells. Well, I thought this goal should be possible, at least in theory, because of two previously established scientific results or scientific streams. The first scientific stream was, of course, initiated by Dr. John Gurdon. So as you heard from his talk, Sir Gardner showed that yes, we can reprogram somatic cells back into the embryonic state. This is possible. So I was extremely honored to share the Nobel Award with Dr. John Gardner. Well, I respect John a lot because of his science and also because of his hair. The second scientific stream was initiated by Dr. Harold Weintraub. He showed that we can convert cell fates by introducing transcription factor. You know, he identified MyoD, and this single transcription factor can convert mouse fibroblasts into muscle. So because of these two scientific streams, I thought we should be able to convert somatic cells into ES cells by introducing transcription factors. However, I had no idea how many factors are required and how many years it would take to identify those factors. Well, it could be 1 factor, 10 factors, 100 factors, or even more. So it will take, 1 year, 5 years, 10 years, 20 years, 30 years, or even longer. However, very luckily, it took 6 years to identify these 4 factors. So in 2006, we were able to report in Cell that by introducing these 4 factors, we can actually convert mouse skin cells into ES-like stem cells, which we designated iPS cells, induced pluripotent stem cells. And in the next year, in 2007, we and others were able to recapturate the same observation in human. So we can make human iPS cells by the same factor combination. So I would like to use this just one slide to explain how we became able to identify these 4 factors. In the initial 5 years, we spent a lot of time to identify and to collect 24 candidate factors. So many students and many other members of my lab were involved, especially these 3 members of my lab. The second step was essential. We were able to establish a simple but very sensitive assay system. These 2 members, Yoshimi Tokuzawa and Tomoko Ichisaka, did this work. Actually, they made knockout mice of a gene which I thought important in mouse ES cells and mouse early development. However, when Yoshimi, one of my first students, with the help of Tomoko, my very first technician, generated knockout mice, we couldn't see any phenotypes. Knockout mice were just happy. And then I was able to persuade Yoshimi to make knockout ES cells. Knockout ES cells should have a dramatic effect, phenotype. But it turned out that knockout ES cells were just happy. So only the student, Yoshimi, became very unhappy. but it turned out that those knockout mice provided us a very useful system, the simple and sensitive assay system to evaluate these 24 candidates. Then, in 2005, we moved to Kyoto. I became a professor at Kyoto University. So I brought these 24 factors and this simple and sensitive assay system to Kyoto. And I asked another student of mine, Kazutoshi Takahashi, to take over the project. So Kazu tested these 24 candidate factors by using this simple and sensitive assay system. In the beginning, he tested one by one. It was very natural way to do. But we found that any single factor can make ES cells out of somatic cells. Then Kazu proposed another experiment. He thought we were using retroviruses to express these 24 candidates. So he proposed, he said he wanted to mix all the 24 viruses and just put those mixture onto cells. And I thought it was kind of a stupid idea, because just by expressing one gene by retrovirus is not so easy. So combining 24 retroviruses seemed to me and probably to most of scientists not so good idea. But I was very proud of myself, who did not stop his proposal. So he actually did that experiment, and very surprisingly, it worked. So that's how and why we were able to identify those 4 factors. So I should mention again that it was indeed these 3 members of my lab who generated iPS cells. Yoshimi and Kazu were my very first graduate students. Tomoko is my very first technician. So without these 3 members, we could have never generated iPS cells, at least in my own lab. So I'm very, very grateful to these young researchers. So then, I'd like to discuss about potential of iPS cells. There are 2 major medical and pharmaceutical applications of this technology. We can make iPS cells from patients. Then we can expand the number of iPS cells as much as we want. Then we can make many types of somatic cells, like neural cells, cardiac cells, hepatocytes, and pancreatic beta cells from the patient. So then there are 2 types of usages of those patient-derived cells. First, we should be able to generate disease models in Petri dish and use those cells in drug screening. The second application is cell therapy or regenerative medicine. So let me first discuss about the pharmaceutical application. Let me use this intractable disease, ALS, as an example, amyotrophic lateral sclerosis, which is also known as motor neuron disease. Patients suffering from motor neuron disease or ALS, their motor neurons get degenerated. and eventually their motor neurons die. Accordingly, they progressively become unable to move. In the end, they cannot move at all, they cannot speak, they cannot communicate, and of course, they cannot breathe. So without the help of respirator, they cannot maintain their life. However, they can still think, they can still see, they can still feel. So it is a very tough situation, not only for patients, but also for family members. Despite numerous efforts by numerous physicians, we still don't have effective treatments, effective drugs for those patients, primarily because we don't have a good disease model to develop treatments, to perform drug screening. Well, as you know, for many human diseases, we have good animal models. And with the help of those animal models, many drugs have been developed, which turned out to be very effective on human patients. We do have animal models, mouse and rat models of ALS. Many drugs have been developed. Those drugs are very effective on mice and rats. However, very sadly, those drugs are not effective at all on human patients. So that means we do need human cells, human motor neurons from patients to make disease model and to perform drug screening. However, it is impossible to obtain sufficient amounts of motor neurons from patients. So that, I believe that has been a big hurdle to develop effective drugs. However, now with this technology, many researchers in the world have been generating iPS cells from ALS patients, and they have successfully generated motor neurons in a large amount. These motor neurons have the same genetic information with the patient. So now we are trying to make disease models and perform drug screening. In our institute in Kyoto, one of my colleagues, Dr. Haruhisa Inoue, has been working on this approach. So Dr. Inoue has generated multiple iPS cell lines from both ALS patients and also healthy control donors. iPS cells from control and ALS patients, we don't see any differences when they are undifferentiated. But as soon as Dr. Inoue induced differentiation into motor neurons, we observed a marked difference in morphology. As you know, motor neurons have projections known as neurites. And as you can see here, neurites in ALS motor neurons are significantly shorter than those of control motor neurons. So they have only like 50% of length. So we have been using this morphological change, phenotype, to perform drug screening. Now we have collected more than 30,000 candidates, chemicals and natural derivatives, and we have been using this kind of a very simple but very useful robotic system to perform drug screening. And Dr. Inoue has identified some effective candidates, including anacardic acid, a HAT inhibitor, which can revert the short neurites back into the normal length. So we're hoping that in the future we can identify at least one chemical or neural derivative which is effective on human patients. It's still, just a beginning. We still have to do a lot of works, but we very hope we can develop a new drug in the near future. So let me discuss the other application, cell therapy or regenerative medicine. So in Japan and in many other countries, this application has been supported. So in Japan, many clinical trials are actively going on, including Parkinson's disease, macular degeneration, cardiac failure, spinal cord injury, as well as platelet deficiency. I believe one of the most promising applications is the production of platelets and erythrocytes from iPS cells. This is another colleague of my institutes in Kyoto, Dr. Koji Eto. So he can already make platelets or erythrocytes from patients on iPS cells. So these two types of cells are very, I believe, very suitable for iPS cell or based cell therapy, because the biggest hurdle is, of course, safeness. We don't want to see any tumor formation after transplanting cells into patients. That is a nightmare. But these two types of cells do not have nucleus. They cannot proliferate. And we can irradiate these cells before transplantation, before transfusion. That means we can kill all contaminating proliferating cells in advance. So this should be very safe. The biggest hurdle is how we can generate sufficient number of either platelets or erythrocytes to obtain, to cure patients. If we can use, in our institute, we have more than 100 incubators. We estimated that with the current technology, if we use all the incubators we have in our big building, we may be able to prepare a sufficient amount of platelets for one patient. But we have to work much harder. So if we can get help from industries, we may be able to overcome that issue. So, as I described, iPS cells became possible based on 3 scientific streams. The first one is, again, nuclear reprogramming, initiated by Dr. Gurdon. And then Dr. Ian Wilmot succeeded in somatic cloning in mammals, in sheep. And my friend in Kyoto, Dr. Takashi Tada, showed that ES cells have reprogramming factors. The second stream is, again, initiated by Dr. Weintraub, factor-mediated cell fate conversion. And of course, the third scientific stream is ES cells, initiated by Dr. Martin Evans and Dr. Gail Martin. Dr. Austin Smith, my friend, has identified many factors that are essential for the maintenance of pluripotency, including OCT4. And again, Dr. Jamie Thompson generated human ES cells. So without any of these studies, we could have never generated iPS cells. So this last Monday, I received the Nobel Prize on behalf of all of these scientists. And from iPS cells, new scientific streams have already emerged. In 2007, Dr. Rudolf Jenisch provided the first proof of concept of iPS-based cell therapy in mice. In 2009, Dr. George Daly and Dr. Kevin Egan generated iPS cells from human patients for the first time. There is another new stream which emerged from iPS cells. In 2008, Dr. Douglas Melton reported direct cell fate conversion in living mice by introducing a small number of transcription factors. This technology is now called direct reprogramming. And this technology has been followed by many researchers, including my colleague at Gladstone Institute, Dr. Deepak Srivastava, who provided the first proof of concept of direct reprogramming-based therapy in living mice earlier this year. So I really hope in the near future, these new technologies will indeed contribute to human patients suffering from many intractable diseases. So again, I'm very, very grateful to all of these scientists who have advanced the iPSL technology and related technology so rapidly. So I'd like to thank all of my colleagues in two places. One in Kyoto, Center for iPSL Research and Application, which was established 2 years ago. Now we have many employees and students in this building, more than 250. This is only a part of them. I'm grateful to all of them for their hard works. And I also have a small group in San Francisco at Gladstone Institute. I have a much smaller group in San Francisco, but they are all brilliant and independent members of my lab. They have to be independent because I can be there only every 5-6 weeks for only a few days. And very finally, I'd like to express my sincere thanks to my family, including my wife, Chika, and my daughters. In addition, I was able to bring many of my family members to Sweden, including my mother, my sister, my brothers-in-law, my mother-in-law. So this is a very big family trip for us. You know, as you know, being a scientist is full of joys. But at the same time, it's full of stress. So without continuous support from my family, you know, I could have never survived. So I'm grateful to them. And thank you so much for your invitation, and thank you so much for your kind attention. Tusen takk.
Thank you so much. I think we could go on applauding the whole afternoon. But I would like to leave a few minutes for questions and comments. And actually, I will take the opportunity to start. So while you think about what you want to ask, we have some time. So thank you so much for a wonderful lecture. My question concerns the source of generating iPS cells, because most of the studies so far, although alternatives are emerging, are done from skin fibroblasts. And in the long run, maybe you want to go for something less invasive or maybe totally non-invasive. And there are some papers, of course, including your own recent publication, Stem Cells, coming out on peripheral blood-derived iPSC, and even urine-derived human cells for iPSC. Could you comment on these developments, please?
It's an essential question. So the answer is, for us, at least in my center in Kyoto, we have decided to use peripheral blood cells and also cold blood cells as a source of iPS cells for cell therapy. The reason is that we have compared many sources, and we found that iPS cells from peripheral blood and also cold blood, they are of high quality. And of course, it's very easy to obtain those samples from patient, and cold blood are already there. We have many cold blood samples in cold blood bank. So because of those two reasons, we have decided to use blood cells. And we are about to start to generate very fast clinical grade iPS cell lines. We are now recruiting donors. And during this week, I heard from my colleague in Kyoto, they are now kind of interviewing donors. So I think we can start pretty soon.
Excellent. Do we have questions from the audience? Don't be shy. This is... Please.
That's a good question. Well, they have many in common. They have, iPS cells and tumor cells have very high telomerase activity, and of course they are immortal. But, iPS cells, in principle, they do not have any deteriorating genetic mutations. There have been some controversy, but we believe they are free from any tumor-associated genetic mutations. So I believe that's the biggest difference between the two. So I would say tumor cells are immortal forever. However, iPS cells and also ES cells are immortal transiently. So that's the difference.
Okay. Anyone else? Please.
So the technology you developed is extremely efficient and very good, but I was wondering, could it be simplified even more by simply adding extracellular growth factors or more chemicals?
Yeah. Yes. Well, many scientists have been actively working on that approach, including ourselves. But now that we can make iPS cells with plasmids or by Sendai viruses, which are free from genetic integration, we kind of stop working on that project, because once iPS cells are established, they don't have any exogenous factors. It's different from retroviruses. But some other researchers are still working on. And if we can make iPS cells by just one chemical or something like that, I think it's still very good.
Yes, please.
Well, that's what we really want to know. we don't know yet. You know, we have been spending many of our time to answer that question, the mechanism. Not only our own group, but many groups, including, in a sense, including Dr. Garden, has been working on the mechanisms. But, you know, we are now getting some clue, but we still don't have a whole idea or big picture. But once we can understand that, we should be able to, you know, further increase the efficiency and maybe speed up the process. Because right now it takes 3 to 4 weeks to generate iPS cells. And for many, in many cases, 4 weeks is too long. For, you know, for example, to treat patients suffering from spinal cord injuries, we have to transplant cells within like 1 week or 10 days after the onset of injury. So 4 weeks is way too long. So I think we still have to work very hard.
Okay, thank you so much again.
Thank you very much.